Okay. We briefly differentiated between the different types of muscle last class. Cardiac muscle, smooth muscle, skeletal muscle. And for all intents and purposes, skeletal muscle is what we are focusing on in the rest of this section. That's the muscle of choice. That's the muscle that I mean when I say muscle in general. Skeletal muscle that articulates bones, allows movement, etc. We talked about the roles of skeletal muscle. So let's look at the structure of skeletal muscle in preparation for this week's lab, how it produces force. You're going to see how it generates tension in lab, how you can activate it externally through electroshock therapy. Kind of interesting. Structure of skeletal muscle. You'll see skeletal muscle shown here in the background from the outside, top level, looking in. You can see it connected to a bone by a tendon, and there are wrappings and compartments, a lot like the compartments that we saw in the nerve, when we looked at the nerve way back when. So the largest level, the level of the muscle, the whole muscle belly is wrapped in epimysium. Epi, around, epimysium, the outer layer. And within the muscle belly, you have multiple bundles. Those bundles, like in the nerves, are called fascicles, the functional units within the muscle belly proper. And the fascicles are wrapped in perimysium, the perimeter around the fascicles. You can see the fascicles projecting from this image on the left-hand side, perimysium wrapping the fascicles. And we're not done. Within the fascicles proper, there are multiple fibers. Fibers are wrapped in endomysium, and the fibers are the base muscle cell unit. A muscle fiber is a muscle cell. So the base cell unit of the muscle is a muscle fiber. So endomysium wraps the muscle cell, but the border of the cell is the plasma membrane, also known as the sarcolemma. And we'll see that a few times in lecture today. So the muscle cell has an outer boundary, an envelope that contains all of the good stuff within the cell, the cytoplasm, the enzymes, the molecules. That's the sarcolemma. And then that is wrapped in endomysium, a layer of connective tissue that separates the individual fibers. So the fiber is the muscle cell, the base unit of skeletal muscle. Within each muscle fiber, you're going to see a number of what we call myofibrils. It's tempting to think of these like you think of fascicles, or like you think of fibers. They're long, strand-looking, thin filaments within the muscle. The myofibrils are long cylindrical tubes packed into the muscle cell. So myofibrils are contained within the muscle cell. They're not individually wrapped like the other layers of the muscle. So let's take a look at the 3D structure. This is the muscle belly at the highest level. A muscle, if you were to take it out of your body right now, might look like this, and it's wrapped in epimysium around the outside, the entire muscle belly, and the tendon sheath wrapped in epimysium. There's the muscle belly, the whole muscle all together. And then the units, the compartments broken up into the muscle belly are the fascicles. The fascicles are different compartments, divided compartments within the muscle belly that are wrapped in perimysium. And the fascicles themselves are made up of bundles of small fibers. Here called extrafusal muscle fibers, but these are just fibers. And the fibers are the unit of skeletal muscle. That's the cell. The cell is wrapped in endomysium, but it has an envelope or an outer layer that contains everything within the cell. So this is a fiber. This is an individual fiber. The smallest unit that we got to in that last video now takes up the entire screen. You can see the sarcolemma, the plasma membrane, the membrane that contains all the good stuff within the cell. So everything shown here is within the cell, within a muscle fiber. I'm pointing here at the space underneath the membrane. That space is the sarcoplasm, cytoplasm, but sarcoplasm means specifically that in muscle, cytoplasm of muscle. All the space containing all the nutrients, molecules, enzymes, etc., that bathe all the structures within the muscle cell is contained within the sarcoplasm. And the myofibrils are easily distinguishable. Long threads within the muscle fiber cell, and they'll extend the entire length of the cell. These can be 10 to 30 centimeters in length, depending on the muscle. Myofibrils are long. They go the entire length of the muscle cell. There's a number of other interesting structures. You'll see this light blue wrapping the sarcolemma, the plasma membrane, has projections into the cell. Transverse tubules are these also blue projections. They're little holes from the outside in. So anything outside the cell can get deep within the fiber going through these tubules. We call them T-tubules. It's the surface membrane that dives within the cell. This is really useful for transmitting action potentials into the cell. Right? Action potentials spread across the surface of a membrane. In the muscle, that's no exception. The surface of the membrane is the sarcolemma, and if the action potential wants to get at all the bits inside the muscle, well, if it can spread down these transverse tubules, it's got a really good shot of doing that. And these are in close proximity to some other really important structures. One of them I've highlighted already, this yellow mesh lattice work that surrounds every myofibril is called the sarcoplasmic reticulum. This will become more important and obvious as we go through the class. This is where calcium is stored in the muscle. I've mentioned calcium a few times. Whenever I've mentioned it, it's been important. Calcium has been that early warning signal to do something. Well, in the muscle, it's no exception. The sarcoplasmic reticulum is where it is stored. When it's released, something happens. The terminal cisterns are part of the sarcoplasmic reticulum, but they are just right next to the T-tubules. These are what register a signal from outside the cell. This is where the action potential is translated into action inside the muscle cell. That will become clear later on. And as far as the other little bits and pieces go, you might know the mitochondria, the powerhouse of the cell. It's one of Dr. Kane's favorite organelles. The orange convoluted structures here, and then the nuclei. Remember, skeletal muscle is a striated, multinucleated cell. Multiple nuclei as these individual cells fuse together in development and make long muscle fibers. So there's lots of small structures to get to know in the muscle. You can see them all here. Similarly, we can zoom in. This is the muscle fiber. with endomysium wrapping the entire structure. You can see the myofibrils, the stacked columns within the cell. There's the plasma membrane. And I show you this view so you can see the space between all the columns, the space between all the myofibrils. That's where the sarcoplasm sits. It washes over all those structures. And if you zoom in, you can see the T-tubules. The T-tubules are access from the outside of the muscle in to the muscle fiber. You can see the small little holes on the surface there. If I zoom in on the plasma membrane, you can see where something could get into the T-tubule from outside the muscle fiber cell and then spread into the muscle fiber. Those are in really close proximity to the sarcoplasmic reticulum, which wraps around the entire myofibril. Notice it's meshwork over every single myofibril. Calcium must really be important. It must need to be released everywhere to have an effect. Like we said, it's stored in the sarcoplasmic reticulum. We keep it there so that it doesn't have an effect until we want it to have an effect. When we want it to have an effect, it's released and then quickly stored away again. And then you can see the mitochondria spread throughout the muscle fiber and the nuclei on the left-hand side there. So this is the structural arrangement of really important things within the fiber itself, the base unit of skeletal muscle. It's complicated. It is tightly packed. By far, the structure that gives the muscle its characteristic ability to contract are the myofibrils. Those are the things that are turned on and activated. The other structures are support structures. You can see the motor neuron here that will turn the muscle on when it needs to be turned on. We'll see that later today, and then you'll also get to consider that in lab. So the muscle fiber cell is packed. There's lots going on inside. And it's so packed, we'll often redraw it. We'll redraw it in a non-threatening kind of way. You can see all the same structures that we just talked about, but with a lot more space. A lot more space to consider the movement of particles and the arrangement of different structures within the muscle cell. This is a muscle cell, just like before. You can see all the important structures we just talked about, the plasma membrane or the sarcolemma containing everything within the cell. You can see the myofibrils that span one end of the cell to the other. This is just an artist's rendition. It's a squished muscle cell. It's not as long as it would be in practice. It's just fitting on one slide. In the sarcoplasmic reticulum, you don't see the meshwork here. This just looks like a reservoir for calcium, and it is, but you don't get to see the intimate association between the sarcoplasmic reticulum and then all the myofibrils in the cell when we draw it like this. We draw it like this to get it up and out of the way. When we want to understand contraction and what happens, we need to really focus in on the myofibrils. And the sarcoplasmic reticulum gets in the way of that focus. So in this case, we're drawing it up and out of the way. But don't think that these are separate within the cell. We saw in the last two slides they are very intimately related. Here we get to start, we can start to see the striations. We talked about at the whole muscle level, it's striated, it's banded, it's striped. Well, that stripy nature comes from the different gradients or different bands along the myofibrils. All of the myofibrils together give us this striped or striated picture at the whole muscle level. And you can see those stripes as you move from left to right through the myofibril. And then also notice The fibrils are anchored at the ends of the muscle fiber by a protein called dystrophin. Dystrophin. You might have heard of muscular dystrophy. It's a degenerative, terrible disease that's characterized by accelerated wasting of muscle. Muscle wastes away. It loses size and strength, and we can't recover size or strength for those individuals. Muscular dystrophy is related to improper anchoring of the myofibrils. The protein is not structured in a way that allows it to do its job. And so the fibrils start to detach and unravel. And the body says, well, it's not doing its job. We can break these down. We don't need to keep them around. And the muscle gets smaller and weaker. In a normal person, though, a normal healthy individual, we don't really consider dystrophin very much. It simply anchors the myofibrils, and then when you want to contract the muscle, it transmits the force throughout the entire cell. Just an interesting connection with a disease state that you might have heard about. So let's move forward. We want to start to understand the structures that give rise to shortening, that give rise to contraction. The myofibrils are that site of contraction. The cell can change in shape due to the actions at the myofibrils proper. So I'm going to zoom in on one of them on this slide. And what we're looking at is a sarcomere. A sarcomere is the functional unit of the muscle fiber. The functional unit. That is, this is the thing that contracts. The sarcomere shortens. It changes in shape and size. The sarcomere is the area between these two squiggly lines in this case. And these are what are called Z discs. They are the bookends of the sarcomere. And so a muscle fiber made up of these long myofibrils, many of those cylinders stacked within the cell. Each myofibril is made up of repeating sarcomeres. You can see one of them front and center. And then off to either side are the beginning and the end of the next sarcomere. And there would be another one after that, and after that, and after that, the entire length of the muscle. So sarcomeres repeat the length of the muscle, and these are the units that contract and relax, that compress and expand. You can see some different characteristics of the sarcomeres. The sarcomeres themselves are made up of multiple filaments. It's filaments all the way down. Sarcomeres are made up of overlapping thick and thin filaments. And it's these filaments that are activated to contract the muscle. Thick filaments are myosin. The thick bands here, the thick lines, are what we call myosin. The thin filaments, these light thin lines, are called actin. And actin and myosin overlap within the sarcomere. And it's this ability, this characteristic, the overlapping nature of these filaments that allows them to move back and forth. Contract, relax, contract, relax. Myosin is thick and heavy. It's big. Actin is thin and fine. These fibers overlap and they give the sarcomere its striated shape. The stripiness, striations, result from how much overlap there is. So I put these colored rectangles up to show you that when you have actin and myosin, there's a lot of stuff overlapping. It's dense, and the color's dark. When you only have myosin, it's the thick filament. It's not dark, but it's not light. And then over on the right-hand side, where you see just actin, the thinnest fiber, it gives the lightest band. And so these will alternate and repeat depending on how much overlap you have of these individual filaments. That's what gives the muscle its striated, stripy look at the whole muscle level, at the outside looking in. It's the overlap of these filaments within the sarcomeres. There's some other stuff on this slide, but we're not going to touch on it yet. We'll look at it on the next slide. There's a lot of detail here. Just know the sarcomere is the functional unit of the muscle. Actin and myosin are the filaments that overlap. And if you have a hard time remembering which is which, myosin is in the middle. Myosin in the middle. Myosin is in the middle with actin sandwiching it on both sides. But even that is simplistic. We're thinking only in 2 dimensions at this point. I'll show you how this is different when we look at it in 3 dimensions. But myosin is in the middle. Myosin is the thick, heavy filament, and actin sandwiches it between it. Because this comes up in a video that we're going to see later on, I do also want to point out one other honorable mention on this slide, a filament called chitin. It's not involved in contraction. It doesn't overlap, but it's curly like a pig's tail or like a spring. And chitin is what allows the overlap muscle fibers to return to normal. If they slide over top of each other, chitin pulls them back into a resting configuration. It resets the sarcomere. It's like a spring. It allows for recoil or a return to normal. That will come up in the video, so I want to point this out now. You can see where it is in relation to the other fibers. It connects myosin to the bookends, the Z discs at the end of the sarcomere. So you can see the striations a little bit more nicely on this electron micrograph. This is the exact same setup as on the last slide. You can see myosin and actin and their overlap. We're zoomed in on one sarcomere. You can see the bookends, the Z discs of the sarcomere with the corresponding fibers in the middle. Now, the stuff that we didn't talk about on the last slide is highlighted here, plain to see. We try to give some order to the chaos, to name different regions of the sarcomere to help identify them. And we happen to give the name the light staining band the I band. I because the letter I is really skinny, and the filaments within the I band are really skinny. The light staining band is called the I band. It's made only of actin filaments. I'll skip ahead one and contrast that with the band in the middle, the H band or the H zone, if you see it in the textbook, made only of myosin filaments, and the letter H, it's wider than the letter I. It's thicker than the letter I. It's made only of the thicker filaments. Of the two, actin and myosin, actin is thin, myosin is thick. The I band, I is skinny, contains only skinny filaments. The H band, H is wider, contains the wider filaments. So the I band and the H band differentiate them by the size of the letter that describes them. And meanwhile, the zone that I glossed over, the A band in the middle, is the zone where the action happens, where contraction occurs, where the filaments will slide and move in relation to each other. This is the band that will change in size and in shape during contraction. We'll see that as the muscle contracts coming up. So I band, A band, H band or H zone, just ways to describe the striations of the different areas of the myofibril. And since we did it on the last slide, if I were to overlay the colors, the bands, the stripes or the striations, it would look something like this. And you can imagine this sequence repeating over and over and over and over again. And if you zoomed out far enough, it would just look like stripes or striations at the whole muscle level. This is where the striations come from. Dark staining when both filaments are there, medium when it's only myosin, and really light staining with the lightest filament, actin, in the I band. Now, in both cases, I think I've probably beaten a dead horse enough. You know the arrangement of the filaments. But in both cases, we've considered them only in 2 dimensions. Actin and myosin sliding over top of each other, back and forth like an accordion. But in practice, it couldn't be more different. Instead of each myosin being in contact with 2 actin filaments, one on the bottom and one on top, it's in contact with 6 actin filaments. Every myosin filament in the middle, the big, thick filament, is in contact with 6 actin filaments spread evenly around it in 3 dimensions. So I've taken one myofibril, well, I haven't, the researchers did. In each of these cases, these are myofibrils at different stages of contraction, but otherwise they are myofibrils. And we've cross-sectioned it, a transverse section through the myofibrils. So you're taking the cylinder, it's coming out of the screen at you, and you're looking at it head on. And you can see the thick myosin filaments, these larger O's, these larger circles, surrounded by 6 thin actin filaments at any given point in time. In practice, in 3 dimensions, there's a lot more to the picture than we put up on the last 2 slides. And this is really good. We'll see why this is good when we start to understand contraction. Contraction. How is the muscle stimulated to contract? How is the muscle recruited? Recruitment is another word for activation, turning on, initiation of contraction. How is contraction achieved in skeletal muscle? A normal resting configuration has the actin and myosin filaments sitting in the myofibril. How do we recruit them and activate them to force them to contract? What is the signal to turn on the muscle? If you know how a signal is transmitted through a synapse, you know how the muscle is activated. Synapses, from week 4, when we talked about action potentials, are coming back to haunt you. If you know how a signal is sent via a synapse, you know how the muscle is recruited. This looks familiar. It's a big wall of text, but as you read through it, you see some familiar players. A nerve sends a signal, and at the end of that nerve, at the synaptic end bulb, voltage-gated calcium channels open, We reviewed this last week quickly when we talked about the common mistakes on midterm 2. Yeah. Voltage-gated calcium channels open. Calcium rushes in. The neuron is prompted to release a neurotransmitter in response to calcium rushing in. That neurotransmitter goes into the synaptic cleft, the space between the presynaptic neuron and then whatever comes next. Until now, that thing that comes next has been another neuron, but now it's the muscle that comes next. The neurotransmitter is released into the synaptic cleft. It binds to a ligand-gated sodium channel on the muscle, on the sarcolemma or the plasma membrane of the muscle, which allows sodium to rush into the muscle. You'll remember, muscle, like nervous tissue, is excitable. It can be activated. It can generate an action potential in muscle. And sodium rushing in does just that. It depolarizes the membrane. An action potential is generated, and it spreads across the surface of the muscle cell. All the events that you know in the propagation of an action potential down a nerve occur here. Sodium rushes in, depolarizes the muscle cell, and spreads across the surface of the membrane. Now is where things start to get different, where things diverge. In the muscle cell, depolarization does something. It doesn't just spread. Depolarization does something. That something is release of calcium from the sarcoplasmic reticulum. So as soon as you trigger or activate or recruit a muscle, the first step in whatever the muscle does is calcium gets released. We're going to leave the rest of that as a black box for now, but calcium is released in response to the action potential spreading across the membrane surface. And then the neurotransmitter is broken down by acetylcholinesterase, or it's taken back up into the neuron. It's removed from the synaptic cleft so that you don't continuously try to activate the muscle. That's how the signal is sent and the muscle is recruited. So you have this information in the slides. You have a flow chart or a diagram like this that lays out everything that I just wrote on that last wordy block of text slide. You can follow these numbers through and you'll come to the same conclusions. This puts pictures to the words that I had on the last slide. And I'll do you one better and show you a quick video that hammers this point home nicely.
Movement in the body is caused by skeletal muscle contraction, which is muscle shortness. Nerve impulses originating from the brain or spinal cord excite skeletal muscle cells to initiate muscle contraction. A group of muscle cells stimulated by a single motor neuron is known as a motor unit. There can be many motor units in a whole muscle. Each muscle cell consists of many rock-like myofibrils that contain the contractile proteins of the cell. To cause muscle contraction, the motor neuron must initiate an electrical impulse in the skeletal muscle cells, even though the muscle and neuron cells do not touch. There is a gap known as the synaptic cleft between the neuron membrane and the muscle cell membrane that only a chemical signal can cross. The neuromuscular junction is this chemical synapse between the motor neuron membrane and the muscle cell membrane. Once the electrical impulse arrives at the end of the motor neuron, voltage-dependent calcium This increase in intracellular calcium causes the release of neurotransmitter acetylcholine into the synaptic cleft. At the muscle cell membrane, known as the sarcal level, the neurotransmitter attaches to chemically gated sodium channels, causing the channels to open. This allows the passage of sodium into the skeletal muscle cell. The entry of sodium creates an electrical impulse, also known as an action potential, which excites the sarcolemma. Once an action potential is propagated along the sarcolemma, any remaining acetylcholine in the synaptic cleft is broken down by the enzyme acetylcholinesterase. At this point, action potentials will stop being produced by the sarcolemma, and excitation of skeletal muscle cells will end.
I was really trying to find a way to make it louder, but I didn't want to interrupt you halfway through. Thank you for all being attentive and quiet, allowing the quiet speakers to do their job. So at this point, we have an idea of how the signal is sent, how the muscle is recruited. We don't know how that signal is translated into movement. Those two are not automatically connected. Right now, we're halfway through this other very informative flow chart that you have in your notes that lists the steps connecting the signal with contraction. So far, we've only gone through these first few points on how the signal is generated. The nervous signal is sensed by the muscle and calcium is released. But that doesn't automatically mean contraction will occur. In practice, every single time this happens, calcium being released, contraction occurs. In practice, those two things are synonymous. But the mechanism is not automatic. How does the muscle then contract? It's a somewhat difficult concept to connect the idea of action potentials to calcium release to filament sliding and contracting, generating force. I'm not going through this in detail. This is, again, something that I included in the notes that's on Moodle that you can use to try to understand the sequence of events. You can picture the motion as it occurs. You can see the neuron delivering a signal. It's spreading through the T-tubules, which is really nice. Calcium being released from the sarcoplasmic reticulum, and then, of course, the accordion-style contraction of the sarcomere. So review that at your leisure. We are going through a much drier approach in class. How does contraction occur? Myosin and actin are central to contraction in the muscle. Myosin specifically is central to contraction in the muscle. Myosin is the protein that uses energy and makes motion. Myosin is the protein that moves, that contracts. It essentially pulls on actin. Think of it kind of like climbing a rope in the gym, which we never really do in the gym anymore, but in old-timey PE class, you used to climb a rope and hit a flag at the top and try to climb down without falling. Think of it like climbing a rope. Myosin climbs the actin rope during contraction. So myosin, shown here, central to skeletal muscle contraction. And the shape is somewhat important as well. Each myosin molecule, you'll see it in another video coming up, has two heads and then tails that are intertwined, and those tails intertwine with other tails of other myosin molecules that make up the entire filament. So think of it kind of like a pile of straws. The straws that have those flexible necks. And the necks are sticking out in all different directions. And the necks are the things that have the binding sites for actin and are trying to climb the actin rope. Myosin, really important in contraction. What's not shown here are two proteins that are permissive in contraction, that allow contraction to occur. Maybe more importantly, they stop contraction from occurring when it's not supposed to. They block important aspects of the myosin-actin scaffold or binding sites. These are regulatory proteins called troponin and tropomyosin. We'll see those coming up. And they are not the only proteins within the muscle. There are other structural proteins that make sure any tension or force that is produced gets transmitted through the entire muscle fiber, that actin and myosin stay arranged properly, that the filaments are lined up properly. I'm not going to ask you to repeat the list of structural proteins, but notice ones that we've talked about already. Titin is on here. That helps the sarcomere to recoil. And dystrophin, that anchors the myofibrils at the ends of the muscle cell. Those are structural proteins that are included in the list. There are others. We're not going to talk about them in detail. We will talk about actin and myosin in detail. And troponin and tropomyosin. Let's get going. So notice the structure of myosin. And there's a couple of really important features. Actin binding site and an ATP binding site. These are really important characteristics of myosin. You know ATP, adenosine triphosphate, the energy currency of the cell, right? ATP is broken down. We break phosphates off to harness energy, and that energy allows us to do something, to do work. Myosin binds ATP. Myosin also binds actin, and it has the ability to work as an enzyme. So that straw within the filament is also an enzyme. It's not just a protein that moves, it has enzymatic activity. What does it do as an enzyme? It breaks down ATP. Breaks the phosphate off to harness energy. You'll hear more about that in biochemistry as you get there. For right now, we just need to know that myosin has three important characteristics. It binds ATP, it binds actin, and it can break ATP apart, harnessing energy. Well, we're not exactly sure why it would do that yet. How is myosin prompted to bind actin? How is it prompted to bind ATP? How is it prompted to break down ATP? I'm going to skip ahead of why it happens for a moment. I want to tell you what happens when it's told to contract. The action of contraction is the accordion movement of the filaments, or the sliding filament mechanism. We just talked about the signal. Nerve sends an action potential that spreads through the T-tubules and releases calcium. There's something that connects that with the filaments sliding over top of each other. That will become clear, but let's just consider contraction for a minute. Contraction is shortening of the muscle, generation of force or tension or pull within the muscle, and it's a result of the sliding filament mechanism. So actin and myosin slide over top of each other. Myosin climbs the actin rope. It does that the entire length of the myosin filament. It pulls all of the actin it can possibly get its hands on. So the entire sarcomere compresses. The Z discs, the bookends, are drawn closer together. The proteins themselves don't actually move, or they don't actually change shape. It's tempting to think that the compression of the sarcomere means twisting of the proteins, squishing of the proteins, but they don't actually change in shape appreciably. They slide over top of one another. And the other structural proteins simply make sure that that force is transmitted linearly through the entire myofibril. So the filaments slide over top of each other. That doesn't just happen on its own. The action of sliding is the result of something specific that happens with myosin. We're going to come back to class on Thursday and talk about that in a fair bit of detail. So have a wonderful afternoon. Class is over for today, and I'll see you Thursday morning.